Hello everyone. Today we will learn about antigen. What are antigens? What are different types of antigen that I have already discussed and put it in form of a video. It is available and I will putting the link of the same in the description box also. So it is very important to learn those two questions, what are antigens and what are different type of antigens, before going through this video. This is just in continuation of the first one. So now, after knowing what are antigens, we have to know two properties associated with antigens. One is immunogenicity, another is antigenicity. Talking about immunogenicity, it's the ability of a substance to induce cellular response as well as humoral response. So molecules or substances that can induce both kind of responses. What a cellular response? That will be the cells of immune system will respond. If you want to know more about cells of immune system from me, you can go to my video which I have put on this on the topic cells of immune system, and I will be sharing the link in this video also of the same, that is of immune cells. And humoral responses, activation of B cells. Activation of B cells is an interesting topic in itself. We will be learning it soon. Here, just remember that immunogenicity is the property of or the ability of a molecule to induce both kind of response, that is cellular and humoral. So immunogenicity will be possessed by immunogens. And what are those immunogens? Are foreign particles. They will be foreign to the host. Then their chemical composition will be a bit complex. They will not be the repetition of the same thing, though sometimes some carbohydrates can also behave as immunogens, but most of the time proteins are good immunogens. Antigenic and immunogenic, and I will talk about what is the difference between antigenicity and immunogenicity in the next slide. So here you remember that proteins are most antigenic and immunogenic than polysaccharides, lipids, and nucleic acids. Even one more property of immunogen is they are large molecules with molecular weight more than 10,000 Daltons. Exceptions are always there. Even some smaller molecular weight compound do behave as immunogens. Now coming to next property, that is antigenicity. Antigenicity is ability to specifically combine with final products of immune response. What are the final product of immune response, and what is specifically if they combine? What is specificity in this, and what are the final products? We will learn in the next slide. In this slide, you can see that whenever an antigen enters the host, it activates its B cells. If it is having antigenicity, if the property of antigenicity is there, it will activate B cells. Once B cells are activated, they will differentiate into plasma cells. Once plasma cells are there, they start secreting immunoglobulins. And the same immunoglobulins then will combine with the antigen, which has activated the B cells. And that same antigen is on the surface also of all the surface of the antigen will be having epitopes on it. Epitopes are the chemical binding sites of antigen. That means they are the receptors. They can bind with paratopes of antibodies or immunoglobulins. So epitope are antigenic determinant. They are the unit of antigenicity. That means number of epitopes will determine how antigenic this molecule will be. An antigen can produce number of these determinants. That means an antigen can have n number of epitopes on it. And each type of epitope will combine with a specific antibody. So specificity is also there. I have already discussed how many types of antibodies are there, and they are how they bind with antigen. That you can see in my previous videos is there when I'm talking about antibodies. So here I think epitope is clear. That is the unit of antigenicity, and it's a binding site where a paratope of antibody and epitope of antigen binds. It's a lock and key kind of arrangement. A specific key will go to a specific lock. So that was about antigenicity and immunogenicity. I hope antigenicity and immunogenicity is clear. In case of immunogenicity, immune cells as well as antibodies are produced. While in the case of antigenicity, only antibodies will be produced, and antigenicity will be defined if antigen is combining with antibodies. While every immunogen is antigenic in nature also, because it is producing both kinds of response, cellular as well as humoral, while antigenicity is when only humoral response is produced. So all antigens having antigenicity will be immunogen also. I hope I am clear now. Now coming to an antigen, which is a complete antigen, which is an complete antigen, substances that can induce antibody formation by themselves. That is, they are capable of activating B cells and making them differentiate into plasma cells, and again then they are combining with specific antibodies. These are complete antigen, when they can do their whole work by themselves. They don't need any help. And then they will react specifically with antibodies too. There are incomplete antigens also, which are known by the name haptens. Haptens are the molecules that can trigger immune response only when combining with a carrier protein. They need help. They will ask for a protein. A carrier protein will be there. If that carrier protein is there, then this hapten will bind to that carrier protein and then start the whole process that I have shown you, how the antigen activates the B cell. And that hapten carrier protein together, that complex of hapten and carrier protein together, is called an adapt. So this was all about antigens. In the part 2, there are more interesting things remaining about antigens, which we will be discussing in part 3. Thank you for being a patient listener.